Hello students, welcome. Today we are going to do Form 1 Topic 3. The name of the topic is called Chemistry of Acids, Bases and Indicators. We are going to start with the Lesson 1 Introduction. We are saying some of the foods that man eats are sour while others are bitter. So the sour taste is due to the presence of acids, whereas the bitter taste is due to the presence of bases. So next, let's see examples of substances that contain acids. They include fruits such as oranges and lemons and sour milk. Then examples of substances that contain bases include antacid tablets and wood ash solution. These are only some examples of the food substances that contains acids and bases, and we have many other examples. So next we are saying using taste to classify substances as acids or bases is not an accurate method. There are, however, certain substances which show different colors when in acids or bases. Such substances are called indicators. So next we are saying definition of acid and base using taste. During the exam, you can be told distinguish between acid and base using taste. So an acid is a substance that has a sour taste, while a base is a substance that has a bitter taste. Next, we are going to proceed to indicators. What are indicators? We are saying an indicator is a substance that gives a definite color in acids and a definite color in bases. So next, what are the types of indicators or categories of indicators? We are saying there are three types of indicators. Number one, we are having simple acid-base indicators. Number two, we are having commercial indicators. And number three, we are having universal indicators. Each of these type of indicators will be discussed in details. So let's start with the first type of indicator, that is simple acid-base indicators. And we are saying a simple acid-base indicator is prepared in the laboratory from flower extracts, red cabbage, tradescantia, and beetroot. So next, let's see description on how flower extract is prepared in the laboratory. So we're going to say crush the flower extracts using mortar and pestle, then add a small amount of ethanol or borobanol. Then we are saying continue crushing until a deep colored solution of the extract is obtained. Then decant the solution into a beaker or a boiling tube. Then we are going to use the extract that we have obtained to identify substances as either bases, acids, or neutral. How are we going to do that? We are going to take 2 centimeter cube of some acid solutions like hydrochloric acid, like sulfuric acid, like ethanoic acid, and 2 centimeter cube of bases of like sodium hydroxide, ammonia solution. And we can take 2 centimeter cube of neutral solutions such as sodium chloride and also water. Then we are going to take some 3 drops or maybe 5 drops of the extract that we have prepared. Then we put into those 2 centimeter cube separately. Then the ones that are acid, all of them, they are going to have a color, or they show a color to that extract. Then the ones that are bases, they are going to have again another color, but different from the color of the base. Then the ones that were in neutral solutions, they are supposed to have no effect on that flower extract. So next, let's see advantages of using simple acid-base indicator like the flower extract. We are saying it is cheap and readily available. Next, let's see the disadvantages of using simple acid-base indicators like that flower extract indicator. We are saying number one, color of the flower extract solution fades after some time. That means The best result for flower extract should be used when freshly prepared. Number 2, it gives inconsistent results. That's the two disadvantages of using flower extract as a simple acid-base indicator. So let's proceed to the second type of indicator. It's called commercial indicators. We are saying commercial indicators are manufactured in industries for commercial use and are commonly used in a school laboratory. As they give consistent results, that's the first advantage of using commercial indicator, and can last for a long time without changing. That's again the second advantage of using commercial indicator as acid-base indicator. So what are the examples of commercial indicators? We have 3. Number 1, litmus solution indicator. Then this litmus solution, we can have it in a paper form. It's called litmus paper. It is of two kinds. We're having blue litmus paper and we're having red litmus paper. Number 2, we're having phenolphthalein indicator. And number 3, we're having methyl orange indicator. So let us see effects of the commercial indicators on acids, bases, and neutral solutions. So we're going to have the indicators here in this column: litmus indicator, phenolphthalein indicator, and we're having methyl orange indicator. The color they show in acid, we are going to have in this column, base this column, then neutral this other column. So if we're having litmus paper, it will show red. So if the litmus paper is red, it will remain red when we place in acid solution or acidic solution. If it is blue litmus paper, it will turn red. Then if we are dealing with base, like maybe sodium hydroxide solution, when we put red litmus paper, it turns blue. And if we put blue litmus paper, it remains blue. Then the neutral, we are going to have bubble when we are dealing with litmus solution. But if we are dealing with red litmus paper, in neutral, the red litmus paper remains red. Then if we are dealing with the blue litmus paper, in neutral solution, it remains blue. That's why we are saying neutral substances, they are neutral. That means they have no effect on the litmus paper. Then next, we are going to go to phenolphthalein indicator. When we put it or we put it in acids, it is going to have colorless. Then when it's in base, it shows pink. Then when it's neutral, it becomes again colorless. Methyl orange indicator, when we put it in acid, it becomes pink. When we put it in base, it's becoming yellow. Then when it's neutral, it will become orange. So phenolphthalein indicator is colorless. So that means when we put it in neutral solution, it remains colorless because it has no effect. Neutral solutions have no effect on the indicators. Then methyl orange is orange in color. So when we put it in neutral solution, it remains orange. So next, let's proceed to various solutions classified as acidic, basic, or neutral solutions. So here we're having acidic solution, basic solution. These are neutral solutions or substances. So we're having here acidic solutions, orange juice, we're having lemon juice, we're having soy milk, we're having vinegar, we're having hydrochloric acid, we're having sulfuric acid, we're having nitric acid, we're having ethanoic acid. So all these are acids. So what about the basic solutions? We're having antacid tablet solution, wood ash solution, we're having toothpaste, we're having soap solution, baking powder solution, we're having sodium hydroxide, calcium hydroxide, and ammonia solution, which is also known as ammonium hydroxide. In neutral solutions, we're having water, it's a neutral solution, then we're having sodium chloride. This water is distilled water. If you are having rainwater becomes acidic solution. You have to know that. Then how do we know if substances are acids and if they are bases? So the ones that are acids, most of them, they are having the word acid at the back, like hydrochloric acid, sulfuric acid, nitric acid, ethanoic acid, phosphoric acid, benzoic acid. All of those ones, they're having acid at the back, carbonic acid. There are very many. The ones that are bases or basic, they are going to have oxide or hydroxide at the back. For example, we're having sodium hydroxide, calcium hydroxide, ammonium hydroxide, magnesium hydroxide. All of those ones are bases. Then oxide, like for example, we can have magnesium oxide, we can have calcium oxide. All of those ones are again bases. So if you want to know a base, you check the last part of that substance. If it's having oxide or hydroxide, you have to know that it's a base. So next we are told, note the following. Number 1, the advantage of using methyl orange over phenolphthalein indicator is that it gives different distinct colors in acid and neutral solutions, while phenolphthalein indicator does not, because phenolphthalein indicators shows both acids and neutral solution colorless. But when we're talking about methyl orange, it shows pink in acids and orange in neutral solution. So it's having two distinct colors. But phenolphthalein indicator does not show those two distinct colors. So next, number 2, we are saying the advantages of using commercial indicators over simple acid-base indicators is that, number 1, commercial indicators give consistent results, unlike the simple acid-base indicators. Number 2, we are saying commercial indicators can last for a long time without changing, unlike simple acid-base indicators. So next, let's proceed to the last part of our class, that is the extended questions. We have 4 extended questions. That's the end of our class for today. Thank you for watching.